All right, we're going to go ahead and start right on time. So welcome everyone to Medical Grand Rounds at the University of Colorado. To our house staff who are here with us on the first floor of AIP2, thank you. I appreciate it. It's good to see everybody in person. As a little teaser for folks who are considering coming in the next couple of weeks, on October 18th, we're going to hear from our very own Dr. Jane Roosh, who is a professor in the Division of Endocrinology, and she's going to talk about exercise, diabetes, and longevity. And then on October 25th, we have a special guest coming, a person that many of you may know. Dr. Kimberly Manning is going to be joining us from Emory University. She'll be giving grand rounds. She'll also be speaking at our Mentorship Academy on revolutionary drivers of change using narrative medicine. As a reminder, CME and MOC credit is available for all grand rounds this year. I will take questions from the live audience primarily, but our chief residents will also monitor the Zoom Q&A feature. And now I am very pleased to welcome today's speaker, Dr. Tracy Bale. Dr. Bale is a professor in the Department of Psychiatry at the University of Colorado School of Medicine. She is also the Anschutz Foundation Endowed Chair in Women's Integrated Mental and Physical Health Research. She's the Director of Intergenerational Stress and Health, and she's also the Director of the Department of Psychiatry Program in Sex Differences Research. She did her undergraduate work at Washington State University. She got her PhD in neurobiology from the University of Washington, and then did her postdoctoral fellowship at the Salk Institute under the venerable Dr. Wiley Bale. Dr. Bale spent her early faculty years at the University of Pennsylvania. She was there for 15 years, I believe, before moving to the University of Maryland. And in 2022, we were lucky enough to recruit her here to Colorado. She's a highly awarded scientist and investigator and a tireless mentor and educator who currently holds 3 R01s and an R37 from the NIH. Her work on stress and trauma has broadly resonated across many scientific disciplines, which I'll highlight here in a second with her awards, but also really across the public sphere. In recent years, she's been awarded the Joseph Erlanger Award from the American Physiological Society, a Career Development Award from the Society for Neuroscience, the Weitz Memorial Award from the Endocrine Society, and a Ziskin-Sommerfeld Research Award from the Society of Biological Psychiatry. I went deep on the CV. I also know that you were named one of Maryland's top 100 women in 2021. Quick Google search tells me there were 3.2 million women living in Maryland at the time. So that's pretty good. Her research focuses on the understanding of mechanisms by which stress and trauma impact brain development and function across the lifespan. Her lab examines the basic underpinnings of these mechanisms, but also their impact in humans, attempting to translate their findings to identify those processes and biomarkers that are important for promoting disease, risk, and resilience, especially in vulnerable populations. As a director, Dr. Bale works to engage the community, developing collaborations and partnerships with local organizations, health officials, social workers, and policymakers, partners with local government, schools and staff, families, and community leaders to provide a lens through which policy, education, and community can be viewed, focusing on the lasting and significant effects across generations of trauma, discrimination, and violence. It truly is a pleasure to welcome Dr. Bale.
Okay, hopefully everyone can hear me. Yeah, great. Thank you for that exciting and comprehensive introduction. I hope the message that comes across is that both I am a neuroscientist in the Department of Psychiatry, but that my focus is really quite translational. And I hope that that's sort of my goal with focusing on the EVs today that I'm going to tell you about. I also want to say that we're super excited to be here. So my spouse, Scott Thompson, is also a neuroscientist. We were both recruited as part of Neil Epperson, who is chair of psychiatry. Part of her goals to build the psychiatry department to be much more translational and engaged on campus. So I hope after my talk today that you will see, because I know we have endocrinology here, we have pulmonary here, we have an array of backgrounds. So I hope that I have lots and lots of emails that follow up after my talk about ways we can collaborate. I will also highlight, although if I say it, you're not going to hear anything after I say this, but Dr. Thompson's work is on psychedelics. Usually when I say that, everyone shuts down and they all get excited about I'm not talking about psychedelics today. That's his area. You can talk to him. So I decided that to give a background around what extracellular vesicles are, first of all, and to really capture, because I know, again, we also have career diversity here, I want to thank individuals who are on the Zoom. I can't see how many there are, but I am thankful for you attending in person. Post-pandemic, I know we all struggle a little bit with sometimes doing the in-person thing. Lunch seems to be a great enticement. I used to say it was a great enticement for graduate students, but quite honestly, I think it's a good enticement for faculty as well to have a free lunch. Please interrupt at any time if you want clarification. I, again, am a PhD. I sometimes use jargon and may not explain everything to the point that you would like, and I am not offended at all if you ask for explanation or want to back up. I would rather we walk away understanding, and if you wait till the end, you may lose out in between, so please do not hesitate to interrupt. It's not a big deal. Okay, so slides are not advancing. Anybody have any thoughts? Mouse, maybe? No? There, okay, we'll just do it that way. That's fine. I'll figure it out. Don't worry. Thank you. No, we're good. So I was told I need to put this up. So does anybody need, you guys need time to scan this? Yes? Okay, I'll wait a minute. Everybody got it who needs it? Okay, great. Okay. Okay, great. So the main focus of my research for the last several decades has been as a neuroscientist in fitting into the translational psychiatry space is stress. And this is going to be something that no matter what your career focuses on, we all understand. I don't care what career stage you're at. Stress is very common, especially in academia, academic medicine, absolutely. You have chosen a stressful career path, likely because you're passionate about the care and the research and the focus of your area of research, but stress itself on the right background can be very engaged, can be sort of provocative, it can be motivating. How many people wait to the last minute to turn in a grant or a prospectus or a whatever, right? Because you need that motivation. That's not the kind of stress we're talking about. The kind of stress we're talking about is when it's on a background of other trauma or other genetics that puts you at risk for neuropsychiatric disease. The stress that is pervasive, compounding, and chronic, and nowadays, it's not hard to imagine. We have our politics in this country, incredibly stressful. I'm the president of the International Brain Research Organization, which means I travel internationally a lot. There's not a country that I have been to, not an Uber driver that I've been in a car with who has not wanted to know what the heck is going on in the United States. It's incredibly stressful, comes across your screen all the time. Family members, it's all very, very stressful. You add on top of that, especially for our younger career stage individuals in the audience who grew up with social media, also can be incredibly stressful. It's a way that we connect, but it's also a way that makes us feel like we're not doing enough. Everybody has a grant, everybody has a position, everybody has a career path. It's sort of what it can feel like when you're a little bit wandering. So that can be incredibly stressful. A global crisis right now with climate change, hugely stressful. I don't care where you live. This is adding, there's a whole field now of psychiatry, really, that is focusing on climate change. It's a big stressor, again, that we're all facing. And then you add in the pandemic. Now, speaking as a neuroscientist, the pandemic is incredibly stressful for all of us. It was unprecedented. We'd never experienced something like this. We didn't know if our family members, our children, et cetera, were safe. We didn't know how to act. We didn't know where to go. You all, especially involved in medicine, extra stress. The social isolation that especially happened for, if you think about the lifespan, more of our senior citizens who were living alone, but especially, I know I had graduate students in the lab who lived alone, and during the peak of the pandemic, interacted with nobody. And I will tell you as a neuroscientist, the impact of that is not going away. And the evidence for that is if you look at the CDC's, the increase in anxiety, depression, or anxiety and depression, again, this is just in adults. The statistics are even more astounding if you look at younger and older populations. So the yellow line, it's across each one of these from the CDC is where we were before the pandemic. So you can see somewhere between 6, 8, 10% presenting with these symptoms. This is as of, the purple bar is as of December 2021. I have not swapped this out. There are new data that go as far as February of 23. It does not look any different. It's not coming back down. And I will also quickly tell you, I have several grants that involve both preclinical animal models and humans. And one of the ways that we recruit human subjects on campus, which I'll show you in a minute, involves having to bring them in and then do an acute stressor to see how they react, right? You could look at hormone levels, EVs we'll talk about. And one of the things that I have a conversation with Neil Epperson, the chair of psychiatry, is that it's very difficult right now to stress everybody because the baseline is so high. It's hard to bring people in and elevate that. And I'm sure you're seeing, whether it's your colleagues or your patients, that our stress levels, the anxiety levels of anxiety, depression, the number of young adults on this campus who are currently taking psychotropic medication is incredibly high right now. So there's a lot to be considered. I will just say, as a senior investigator, one of the points I'd like to make for my other senior investigators, researchers, and colleagues in the audience is that this is going to have an impact for many years to come on how we evaluate our rising assistant professors, residents, graduate students. It's going to have an impact, and I think we need to be mindful of that. It's not just because this was in the past and now it's all over and we still have to have the same expectations. I think we really need to be thinking, especially for career path individuals who are taking care of family members during the pandemic. It was a huge impact on people's career focus. So I just want to put that out there as a reminder. It's important. We need to be thinking about it. And if we take all of that into consideration, to also be mindful of our community members who are struggling with things like racism, discrimination, bias, poverty, violence in many areas around the Denver area. And as was pointed out, I just came from Baltimore. A lot of this also to be considered, that's adding on to already a high baseline of what people are dealing with. So it's things to think about in terms of how does it affect the brain? What are the coping skills and strategies and the tools that not everybody has? And I think that's so important for how we deal with, maybe how we deal with patients, how we deal with colleagues. Not everybody comes from the same background. Not everybody has the same coping strategies and tools. And so be mindful of that, be patient. This is a slide that just talks about overall how my lab thinks about the research that we do. As I said, I'm a neuroscientist. I consider myself to be a translational researcher. Everything that we do, every grant that I have, so I have the equivalent of 4 R01s. The R37 is basically an R01 that you get lucky as a career award, so you get 10 years instead of 5, which is really nice. But every one of them has preclinical models, animal models on it, and human beings as part of it, right? So we try to go back and forth, and that's what I want to illustrate here, is that we need the preclinical models to get the mechanisms. It's really difficult to assess mechanism in a human population, very difficult. But you can't really understand the value or the relevance of what you're finding in a preclinical model if you can't take it into the human population. So I'm going to give you 2 quick stories just as an illustration of what my lab is focusing on, but keep that in mind. We really like to start often with a finding in human populations that's relevant to how stress affects neuropsychiatric disease, and then work back to the preclinical model, back into the human being, et cetera. So we do across, we have cell culture models, we have mouse models that we work in, we do whole animal assessments, we do all the way down to changes in mitochondria function and changes in epigenetics. In the human subjects, we really try to think about both males and females. We think about the lifespan approach as well. And that includes the preconception environment through pregnancy and across the lifespan. And as was also pointed out in the introduction, we care a lot about the community as well. If you do human subjects research, I think one of the difficulties that occurs around the United States is ensuring that as you recruit somebody in for a part of your study or your clinical trial, how does the information and what you learn get back into that community, right? There's a huge distrust that is built up. It's probably true in Aurora. I've only been here for a year. I haven't spent as much time here, but I will tell you 5 years in Baltimore, I learned a lot about the distrust in many communities of the biomedical enterprise. So we have to make sure that as we're working in our community spaces, informing, engaging, that the information that we learn, we also make sure it gets back into the communities so they know the advantage of participation. That's important. We also do a lot of work working on programs that we built in Baltimore City schools that allowed us to engage with like city council and the mayor's office, et cetera, around mental health. Really important in building similar programs here as well, which I don't have time to talk about today. Okay, so one of the difficulties as a neuroscientist working in this space is how do we think about neuropsychiatric disease? For anybody in the audience who works on cancer or cardiovascular disease or diabetes, if you were sitting with your family members and friends at Thanksgiving and the topic came up, you could pretty easily convince them or discuss with them how you know, what you test for, what improvement looks like. But now think about neuropsychiatric disease, and you recognize it's very difficult to discuss. We don't have biomarkers. Treatments, yeah, some work, some don't. What does working even mean? It requires a really complex conversation with individuals. How do you model that in a mouse? You can't ask the mouse how they're feeling. This is very difficult. So one of the ways that I like to think about this at every grant that I write is thinking back to Tom Insel when he was the director of NIMH. And he liked to espouse this formula, which is the G by E by D. I think it's true for most diseases, but it's really true when thinking about neuropsychiatric disease. The G by E by D, your genetics that you inherit from your parents. That's everything about your risk and your resilience, and there's not a thing you can do about that DNA. That's what you are, right? So maybe your family has a history of hypertension. You know that you need to be careful about your exercise and your diet. Maybe your family has a history of cancer. You need to be particularly concerned about screenings. Maybe your family has a history of mental illness, right? So those are the genes. We know a lot from GWAS about risk, but that risk is only as important as the E. The E is the environment, and that is the environment you're exposed to. What is the diet your mom consumed during pregnancy? What is the diet your family consumes at home? How much exercise do you get? How much stress are you under? All of those kinds of factors. When did your mom have a virus during pregnancy? All of these things matter. None of them are indicative of, yep, you're going to present with schizophrenia. It's the interaction that changes the risk, right? And the D is developmental windows, the when that exposure happened. Did it happen in the first trimester versus the third trimester of your pregnancy? Did it happen to your parents? There was an accumulation of lifetime adversity for mom and or dad before they even conceived you. And how does that change the course of your development? So that's the G by E by D. The bottom there is SABV, sex as a biological variable. And that is so important to all factors of G by E by D, because your sex, the XX of your cells versus so XX versus XY matters for the genes, because there are really important genes that are different between X and Y, and they can matter a lot for how your cells respond to the environment. All right, sex matters for how you interact with your environment, how you perceive your environment across your lifespan, right? There could be interactions with gonadal hormones in puberty. Males and female brain development is very different, right? Males' prefrontal cortex, not fully developed till probably your late 20s, early 30s, very different than a female's necessarily. And it's also dependent on your developmental rates. Even in utero, males and females develop at different rates. The placenta, the XX versus XY cells, if it's a male or a female birth, matter for how the placenta responds to the maternal milieu. All of these things matter. So SABV always matters. And if you've ever submitted a grant recently in the last decade, probably to NIH, you know you need to have a plan for how you will address SABV. So I'm just here to tell you it matters. Okay. So how do we explain the brain, and how do we incorporate this G by E by D into understanding anything about neuropsychiatric disease? The brain is incredibly complicated. I personally think it's the most exciting tissue, but I'm biased. I can't imagine studying anything else, but it is really complicated because the brain in the third trimester is a really different thing to look at than the brain in puberty, right? It's really, really exciting, but very complicated. So I want to start off with one of the things that I mentioned earlier, that we really don't have any biomarkers that really help us understand neuropsychiatric disease. So just quickly, when I say biomarker, it's a spectrum, right? Biomarker can just be, yep, this is a pregnancy that might be at risk. We should watch this pregnancy. To, yep, this is indicating that maybe gestational diabetes is an issue, and this might help us address or intervene therapeutically. Biomarkers kind of encompass that entire spectrum. So extracellular vesicles have come on the scene, and if anybody works on them, especially if you're in the cancer field, you know they've been around for a couple decades. As I always say as a neuroscientist, cancer does all of the work and really sets the ground rules for what something is, and then neuroscience jumps in somewhere in there and really makes it quite sexy. So extracellular vesicles, you may have heard them, sometimes people call them exosomes. Exosomes are a type of extracellular vesicle. I will refer to them as EVs, just to shorten it. These are basically circulating nanoparticles. They're basically lipids, sort of cell-like, but they're not cells, they're teeny tiny. They are secreted from every tissue in the body and in all mammals. And they're super exciting because they're very dynamic. They're very responsive to a changing environment. Suddenly you ate lunch and now your glucose levels are high. You'll get a change in the type and the concentration of EVs. You're starving because your glucose levels are low. You will get a change in EVs. Pregnancy is in the course of the lifespan, a time at which the concentration traveling in circulation goes up 4 to 5-fold. In part because the placenta secretes so many, but also in part because there's a rapid and dynamic need for engagement between the fetal compartment and the maternal compartment, right? So these things are really exciting. They have very specific signals. So I know many of you are in endocrinology. Endocrinology is a great and exciting field. There's a lot of communication happening all the time, but there's a lack of real targeted specificity in endocrinology, right? Estrogen or testosterone acting, you know, it might be important to what acts in terms of enzymatic breakdown of the different steroid hormones. Receptors certainly determine where, but EVs are even more specific in that the proteins in this lipid membrane determine, they can tell you a lot about the tissue of secretion, but they tell you a lot also about the tissue where they're acting, because these things get taken up into cells and they perform a function at those cells. So they can be very local. Imagine a placenta. You have all these different cell types within the placenta, right? And so depending on if it's mid-gestation or maybe late gestation, the placenta is performing different functions, right? Later in gestation, the baby can make a lot of its own products. It doesn't need so much coming from mom. But at mid-gestation, it might need energy. It may need communication. It may need growth factors, right? The way that the placenta can really coordinate that in terms of what's the baby's need or what maybe is happening in mom's milieu, high glucose levels, gestational diabetes, something's not happening, maternal infection, stress. The placenta can quickly coordinate the cell types by secreting EVs that get taken up very locally and can say, okay, here's what we need to be doing functionally in order for the best interest of the fetal development. So they can act locally. They also get secreted into circulation. That gives us the opportunity when taking blood samples from individuals, maybe if we're going to do an acute stressor, so individuals who've been exposed to all kinds of trauma or particular types of trauma, a story I'm going to tell you about in a second. You can get samples. You could say, wow, we're going to give you a resting sort of measure. We're going to get a baseline sample. And now we're going to stress you and then look within subject. And that change can be very informative for where those signals might be coming from and what they might be doing. So what happens when they get taken up? When they get taken up into other cells, the internal composition of EVs, it's proteins also, but it's a lot of small non-coding RNA, especially microRNA. But there's parts of mitochondria, there's all kinds of exciting things going on. Those small non-coding RNA, when they get taken up into other cells, quickly can act. They can quickly, the microRNA can designate specific mRNA and degrade them, right? This happens through the RISC complex of cells. What that means is, in a much faster way than if you have to go all the way back and start with transcription and then get into translation, you can quickly alter a cell's function by shutting down the translation of genes that have been elevated, right? So this is a way, in a very dynamic function that doesn't take the time that transcription might take, that you can change a tissue's function very quickly. So there's two main types. There's many types of EVs, but the two main types we know the most about are exosomes on the left and microvesicles on the right. Exosomes are the very smallest of extracellular vesicles. And as you can see on the left there, the exosomes come from, so two different big differences for these things, the size and how they're secreted from cells and how they act at distant sites. The two things you need to know, exosomes really are designated by the early endosome, and they're packaged into the multivesicular bodies within a cell. Again, all cells, including the brain. And that's how they are secreted outside, right? So they merge with the cell membrane and they are secreted out into circulation or locally. Microvesicles are much bigger in size, slightly different in function, and they are actually take up part of the cell's membrane itself. And that's exciting because if you were to do a proteomics assessment on a change in a population, it might tell you something about the tissue that they're coming from or going to, but especially as they're bringing part of the membrane proteins with them. And I'll show you an example of that in a second. I wanted to put this in here because I knew this was a general audience, that there has been, again, really the last decade has done so much research in this area across fields about how you might incorporate this biomarker into your research. You can get EVs from just about every biofluid. Plasma, you can get a ton, right? But they're coming from everywhere. How do you zone in on more specific function? Well, in lactation, you can get them from breast milk. You can get them from saliva. You can get them from urine. You can get them from semen. I'll tell you a little story about that as well. They have been patented in many areas, and especially around different focuses of different therapeutic use. You can make synthetic EVs, right? There's tons of ways that they're doing this now. I know Penn has a huge program from well over a decade ago when I was at Penn, where they had developed devices in cancer, where they were screening blood plasma for markers of different types of cancers. But they're also making therapeutic EVs where you can be able to target specific sites to treat different diseases, such as cancer, right? If you want a tumor to take up your treatment and not affect the other tissues, this is a really great way to do that. So tons of patents out there. You can also, and many companies are developing ways in which cell culture would give you specific types of EVs. So in our lab, we have, and I'll tell you a story about this in a second, we have epididymal epithelial cells. And I'll tell you in a minute why we're doing epididymis as a neuroscientist. But you can have these cells in culture, and they will secrete pure populations from those cells that you can collect and harvest the EVs. You can also think about ways then to alter them and put them back into patients, for example. A lot of work in the cancer field into this. Just sort of getting into this in psychiatry now. There's a lot of work around neurodegenerative diseases and using them both as biomarkers and drug treatment delivery systems. We're really talking like the last recent year or two that work has been done. So really, really exciting. I will just say that there's been lots of potential biomarkers in psychiatry over the last several decades. Most of them have kind of petered out and not proven to be of real validity. I think EVs are really here to stay. It's been several decades and the data are pretty convincing. There's some data on depression. There's some data on individuals who are responsive to medication and depression. There's still a ways to go, but I think there's some impressive work going on at Stanford right now around neuropsychiatric disease treatments in this area. Okay, how do you measure them? There's tons of information you can get from populations of EVs. One of the most easy accessible, if you're doing a patient population, for example, if we start at higher order assessment, is that you can use a nanocyte technology. We have a machine in my lab. This is using Brownian motion, where you put the samples through, and you can do a pretty high throughput, especially if you have a really energetic graduate student. You can put them through the nanocyte machine. There's all different technologies out there. More expensive will give you more information, obviously, but you can do things like look at the vesicle size. As I just told you, right? Remember that microvesicles are bigger, exosomes are smaller. There's other different types of them. But if you see a shift in your population or within subject following a trigger, a seizure or a stressor or a diabetic insult, et cetera, then you can start to hone in mechanistically, right? And that's a really easy finding to look at. Vesicle size can tell you the types of EVs you might be shifting to, which can lead you down to one path. It can also tell you a little bit about endocytic rate, the changes at the rate at which they're taken up. and the EV type. The zeta potential is something else you can get, right? If you start shifting the protein population and the size in your plasma sample or in your cell culture or whatever you're studying, that's also really informative because changing the zeta potential is a combination of size by charges of all the proteins and structures on your EVs. And that's important because it has to do with the physics of a slipping plane. And what that means is that how these particles are interacting with each other. The more negative the charge overall of your population, the more they're going to bounce off each other and stay in circulation longer. And that tells you something important is happening, that these EVs have a function and they want to stay around longer. The more neutral they might be going to means they may have a rapid action, but they're going to start aggregating and be taken out really quickly. And so that might tell you something functionally as well. So there's stability there. So those are the really obvious and very easy and quite inexpensive things that you can initially do. So if you're doing a new population, you wanted to ask these questions, it could tell you where you might want to go next. If you saw absolutely no changes, then you might want to say, okay, maybe this is not a marker for the question that I'm asking, right? So it's an initial go, no go kind of step. You could also, so I'll also point out, if you wanted to then continue on, you would do things like a proteomics assessment, right? That's a little bit more expensive, depending on how many samples you're doing, maybe a lot more expensive, but you could start to ask questions, and I'll show you some data from that in a minute. You could also harvest your EVs and do small non-coding RNA sequencing. You could look at particles related to mitochondria. There's a number of routes you could then take on a pathway if you found that interesting. So initially, there's a lot of questions that had really not been answered in this field. And I think in part because a lot of this was coming out of cancer, and cancer was really interested in detection and treatment in a way that really this field is lacking in a lot of understanding and development of tools. And so as a neuroscientist and really a neuroendocrinologist, because I study stress, we've been asking lots and lots of questions, both in clinical populations and preclinical models. So I'm just going to touch on a couple of these to give you an idea of how we move back and forth between our models. So first of all, if you look across the lifespan, when would you pick as the most exciting time to really start thinking about EVs and their function? Probably pregnancy. I mean, they go up 5-fold in all mammals. They got to be doing something interesting. And so there's been a lot of work. Yoel Sadowski, who runs the McGee Women's at Pitt, has done a ton of work and how the different cell types talk to each other. But we wanted to know more like in maternal health. These things are secreted in such high concentration. What are they doing towards mom's health? Do they have primary function? And so one of the things my postdoc began doing, Hannah Zierden, who is now an assistant professor at the University of Maryland, College Park. And she works in this she's an engineer from Hopkins who wanted to learn some biology. Very, very savvy career move, because in engineering, they lack people who really understand a lot of biology. And so she was literally plucked right out of my lab and given a faculty, a tenure track position at College Park because she is so talented. So she wanted to ask a question, again, knowing what she knew from clinical settings of maternal gestational diabetes and changes in maternal health with stress, just some primary questions. I'm not going to go into all of the data from her paper, but really just to appreciate in the mouse, similar to the human, across gestation. So that graph on the left, that's from the nanocyte machine looking at concentration across gestation. And as you can see, it goes up really high as pregnancy goes on compared to a non-pregnant mouse, just like the human. And so then she wanted to ask just a basic question of what if we stress mom? So this is mice, not humans. And it's very easy if you don't work in mice to stress them. Why? They are not the smartest creatures in the world. And so it's very easy to do anything different in a mouse in their little cages. Doesn't matter what you do, they get stressed out. So we just do that in a chronic way. Change, novel objects, noise, light, et cetera, olfactory cues, very easy to stress them. So we stress them during the really early, the equivalent of a first trimester pregnancy to the moms. And then we ask the question, what did that do just to the concentration of EVs? And as you can see across gestation on the right there, the y-axis is the concentration. It's significantly reducing them, not just while we're stressing them, long after the stress has ended. So something fundamental about stress, okay, the question would be probably glucocorticoids to start with, and that's exactly what she did next. What did it do? So she began asking just fundamental questions in this model. What happens if it's glucocorticoid mediated? What happens across the circadian rhythm? Mice are nocturnal, so it's reversed from humans. But right, your cortisol levels rise and fall over a 24-hour cycle every single day to basal nadir low levels in the evening, to before you rise in the morning and throughout your early morning day, to really distress levels. That's a normal. Why? Because every cell in your body and in a mouse's body requires a rhythm. And that rhythm is really critical for health. It's critical for sleep. It's critical for your immune response. It's critical for your brain. And so that circadian rhythm is doing something, as you can see on the right there, to intersect with how your tissues are secreting these EVs, which is something that she's now following up on. So if you look at glucocorticoid levels, which is the y-axis on the far right of that graph, you can see when your glucocorticoid levels are low, your EV concentration is high, and throughout the day, that transient changes. Probably in part because glucocorticoid levels are also important for your glucose levels. And so all of that leads me to the point of baseline importance for the concentration of EVs is maintenance of homeostasis in all mammals, right? That's really, really important. Okay, so we've learned a lot about function, just basic primary function of EVs, some of which Hannah's following up with, some of which we're following up with. But I'm just going to end with two really quick stories. One is about trauma, as per the title of this talk. And so this was a collaboration we began doing with Tanya Jovanovic when she was at Emory Hospital in Atlanta, and they have what's called the Grady Trauma Project. Okay, the Grady Trauma is, so the Grady Hospital is their inner city hospital. Kerry Ressler, as an MD PhD in psychiatry, started this program because he recognized all of these individuals coming into the hospital with all different levels of trauma. He's interested in PTSD, but he was thinking, why aren't we capturing all of this information? Men, women, across the lifespan. We should be capturing them, recruiting them back in for different studies. And that's what he began several decades ago. He was recruited to Harvard and now runs the McLean Hospital. Tanya took over the project, and she's now in Detroit because they wanted to start the Detroit Trauma Project. Okay, I'm not going to get into the basics of this study, but we wanted to ask the question about the G by E by D. When a particular type of trauma happened in your lifespan, could we detect a signal decades later in terms of your EVs? And that is a study I'm going to tell you about. So these are an N of somewhere between 25 to 28, mostly African-American women, all in their late 30s, early 40s. We did not recruit them. All of their data was already in the database for the Grady trauma. They pulled it out based on the question, when sexual trauma, and the reason we picked sexual trauma is because it's a dramatic, we know a lot about the impact on health across the lifespan, and it's easily identifiable in your lifespan of when it happened. Okay, so an overall high level of other kinds of trauma in all of these women. The sexual trauma happened before adolescence, in adolescence, as an adult, or no sexual trauma at all. All right, so there's lots of questions we can talk about, but I'm just going to move ahead of what we found. Okay, so quickly, health outcomes. On the left side, what I'm showing you is we're comparing no sexual trauma to all sexual trauma groups overall, and then breaking out the sexual trauma by age of when it happened. And what you can see here, looking at sort of metabolic health, I know Jane Rausch is coming to talk to you soon. Jane and I have talked a lot about this data. This is right in her area, in her wheelhouse. That sexual trauma, really driven by childhood sexual trauma, really predicts risk for hypertension, especially early entry into hypertension, diabetes, and obesity, right? So very metabolic phenotypes. Sexual trauma that happens in adolescence predicted more neuropsychiatric engagement, especially risk for PTSD. All right, so you can look at those two data sets. This is all published. I want to drive home the message that it really did predict different types of health outcomes, but what about the biomarkers? Okay, on the top there in the heat map is looking at unbiased proteomics. We got all of the blood samples sent to us at Maryland from the Grady trauma. We isolated, blinded all of the EVs from all of the blood samples, blinded, sent them to the mass spec facility, blinded, and they sent back all of the data, and then it was uncoded. So what it told us was the levels, so they at the mass spec are able to detect from these EVs all proteins detectable. So you have really, 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 really low abundant proteins and really, really high abundant proteins. Okay, up there on that heat map is showing you from blue to red. If it's blue, it's a really abundant or only abundant in a particular group relative. If it's low, 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 red or pink, it's telling you that it was either non-detectable or very low, right? Okay, so on the top is comparing the two groups, and then below that is breaking out by age. I don't have time to go into all of the exciting things we found here, but just focus on that middle bar there of the breakout. That's the adolescent sexual trauma women, okay? Those blue proteins that are only showing up in only that one group are all proteins that are related to keratins and keratin-like and keratin-associated proteins. As a neuroscientist interested in stress and trauma, my first response was like, oh, that's not very interesting. What does that mean? But then I hearkened back to my postdoctoral days at the Salk Institute and recognizing your skin is your biggest barrier from the environment. Your skin is highly autonomically innervated. And the other thing I remembered from working in an endocrine lab is that the hair follicle in the skin has a very specific stress response itself that has a lot of the same molecules that you have in your brain responding to stress, a lot of the same genes. So then I got more interested. So then I started diving into, and you can see in the dot plot in the middle there, what are all these genes? If you're interested in epigenetics or when things happen earlier in the lifespan or maybe during gestation or preconception, and they're predicted, right? Kaiser Permanente, adverse childhood experiences. Why? How does that happen? What does that mean? So I started thinking epigenetically, what is it about all these genes? What do they have in common? And on the right, you can see what they have in common is that the majority of them are from a gene cluster. And if you study epigenetics, you know, that's great because that tells you they can be regulated in a similar fashion and they can epigenetically be modified depending on a particular time window, right? That they might, the promoter of this cluster might be more open and vulnerable or plastic. So that got exciting. The reviewer for our paper actually said, super exciting, but also, guess what? From GWAS data, 2 other genes are known from PTSD risk, the corticotropin releasing factor receptor 1 and MAPTau, identified for GWAS for PTSD in the same cluster, right? So we're really now diving in human subjects, preclinical models, and trying to figure this out. But what is also very exciting across this is that many of these genes we identified are critical for differentiation of keratinocytes in your skin cells to Merkel cells. And most of you are saying, what's a Merkel cell? That's what I said. Then Artem Patapoutian got the Nobel Prize recently for Merkel cells, right? Merkel cells are very sensitive mechanosensory skin cells. They're in really defined places in all mammals, which in and of itself is kind of exciting. They respond to very light touch. They're in your fingertips. They're in your lips. Well, intimacy, that's a really provocative question, right? Why in your lips? Places on your back, like I said, your fingertips and in your genitals. So the particular type of stress and trauma that we picked, sexual trauma, probably fit a lot into this finding that we came by. The other thing that's very exciting, so the piezo2 is the channel that's responsive for activating Merkel cells in these touch domes. When activated, they are the only neuronal-like skin cell that we have. When you activate them, they release norepinephrine, and they activate the A-beta sensory fibers, the beta-adrenergic 2 receptor on the sensory fibers that then allow that activation to go back up to your sensory cortex. So as a neuroscientist, this opened up a wealth of exciting opportunities because how does this happen? What did the programming do? What happened when this trauma happened? But the finding that really made exciting to write this grant that got funded from NIMH was that Ellen Lumpkin's data, she's a developmental neuroscientist, identified that the final differentiation, the closing of the window of plasticity of these Merkel cells happens in adolescence. So now what does that give us? That gives us the G, 17q21 gene cluster, the E, this particular type of trauma, and the D, the adolescent window. Right? So yes, tons of questions, all kinds of things, but it's really an opportunity that came out of an unbiased approach of these EVs. And now we're doing a grant study with Tanya's group that allows us, she is now in Detroit, she has a whole new cohort that they've been following of women that were recruited in pregnancy, and they followed the kids and the moms for 30 years. So now we have moms, kids, and the next generation as well to ask a lot of questions. We have men and women and all the trauma that they've been exposed to across that 30 years. So stay tuned for that. I'm running out of time, but I want to tell you that what does this mean for preclinical models? There's all kinds of great tools. And even though Artem Patapoutian got the Nobel Prize, no one has really asked the question about what happens when you isolate these things, right? If you isolate them in a way of pressure, isolate them in terms of particular types of trauma, and especially during particular windows of development. And so we have mice, transgenics. It's the only reason we use mice, because otherwise they're not super smart animals, is that because of the transgenics. So we can isolate DREAD GQ, which is a designer receptor exclusively activated of drugs, designer drugs, right? It's a way that Brian Roth has developed a transgene. You can put them in by virus. This happens to be a transgenic mouse we can breed. You can only express them in cells of interest. So we can cross them with the particular Cre recombinase. They're conditional. All you need to know is it means that we can express these in a Merkel cell enriched population. And we can use designer drugs to activate them or inhibit them when we want during pubertal development in a mouse. We can get all kinds of information out about what does that do? What does it mean if you're walking down the street or in a mouse's cage, sitting in your cage, and all of a sudden, out of the blue, your Merkel cells are activated, right? No one's ever asked that question. And I'm just going to point out, that's the cool thing about research, is that you get to be the scientist for the first time anywhere in the world that gets to know what does it mean when your Merkel cells are activated. And I'm going to share with you because Austin Corrigan is a postdoc in the lab that just did this. So I'm not going to tell you which is which. You need to know a little bit about mice. On the left is one genotype, on the right is another. And so some of them have the DREAD receptors in the Merkel cells and some of them don't. They're all going to get the drug. What do mice normally do if you move their cage and you put them in a hood? They climb around, they check stuff out. They're very curious creatures. They're climbing on, etc. So I'm going to turn on these videos and you can see a difference in what the mice do, okay? Watch the ones on the left versus the ones on the right. The ones on the left, you can see kind of climbing around, curious, checking it out. What are the ones on the right doing? They're grooming in their nest. And the common behavior for mice when something's not right, their anxious-like behavior is to groom. And so what happened, the ones on the right are the ones that have the receptor. And so for the first time ever, we've activated Merkel cells in this enriched population in mice, and their response was to be like, this is a little weird. And so they went back and they were grooming in their nests. So that tells us one thing. They didn't freak out. It's not a robust response, but it tells us that they noticed. And that is a really good start to these studies that he's doing. So anyway, I don't have time to go into all of the sperm data, unfortunately, but what I am going to tell you, because this is another grant that we just got funded that actually is with Neil Epperson. I'm a neuroscientist and she's a psychiatrist, and we spend a lot of time talking about sperm. And that is because we are interested in stress and trauma signals as they're conveyed at conception that can alter the course and the timing of how the brain develops, right? That's one of the biggest factors that we don't understand in neurodevelopmental disorders. It's the timing. Does your brain speed up? Does your brain slow down in development? So I don't have time to go into this because I know we're running out of time. But these are data that Neil and I started when we were both at Penn, and we recruited undergrads and medical students to come in every month. So it was a monthly survey for 6 months and included things like final exam stress. And so they did a perceived stress and a whole course of evaluations. We knew about their lifetime adversity as well. And we collected samples each month. And then we took those samples and we paid them heavily. I'm just going to tell you, they got very, very nicely reimbursed for their donations. We sequenced the heck out of their sperm. We wanted to ask the question about all the small non-coding RNA, and that the EVs, so can I go back? Yes, I can. I just want to make sure this is true and this is clear to you in the model. This does not happen in the testes. This does not happen during spermatogenesis. Why? Because would you want something to happen where the environment can intersect with your sperm in the testes? No, you would not. Why? Because you could wipe out a species, right? That's when spermatogenesis is happening and the DNA is very vulnerable. And there's a whole evolutionary conservation of methods of which the testis barrier protects spermatogenesis from the environment. This happens in the epididymis, actually the caput. So the epididymis sits on top of the testes. When sperm exits spermatogenesis in all mammals, they cannot swim and they cannot fertilize. They're not mature. They get pushed into the epididymis and they hang out there kind of in a quiescent state. There's all kinds of really cool mechanisms that the cells use to get the final maturation steps. A key factor in this final maturation are EVs. They're transferring information from the cells that line the epididymis. These are called epididymal epithelial cells, and they present all kinds of information to the maturing sperm that the maturing sperm can then transit the rest of the reproductive tract and carry with them. Okay, so we did this cool study. We wanted to know of the small non-coding RNA that the EVs could be transferring to the sperm. Could we ultimately see any differences if we modeled high levels of perceived stress within subjects, and would those be common across subjects? So the N here is not huge. We ended up with like 22 men that completed all 6 surveys and donations across the months. I think that we really lucked out because we did this at Penn. When we evaluated their overall life adversity, it was almost non-existent. So I think because of that, we didn't have that variance added to it. I'm not going to comment further on that. Ivy League institution, take it what you want. We sequenced all of the sperm. We sequenced the microRNA, the tRNA fragments, and the piRNA. This is just showing you some of what that data looked like. We did a very complex modeling with the bioinformatics and Institute of Genome Sciences at Maryland. It took a better part of 2 years, first to align all of the RNAs, but then to model them, right, with the perceived stress. We knew from our mouse preclinical model that it was unlikely that stress in the month we were collecting the sample was important, but rather it was the prior stress, right, when stress resolves. Think about baby boomers after the war has ended. Think about fertility issues after a family adopts, right? It's after the stress is over that you see this change in fertility. So we were interested in sequencing them. This just shows you the modeling. Some really, if there's any reproductive biologists or endocrinologists in the room, some really interesting cyclic differences that we still are following up on because that may be related to fertility too. That is unknown for men. Anyway, I'm just going to quickly tell you, along the microRNA route, identified some really top candidates, 2 of them known importance for conception and implantation. So very exciting, very validating. But we identified the time as being somewhere between 2 and 3 months after high perceived stress. Why am I telling you that? Because we've now followed up on this in our preclinical models. A prior graduate student, Jennifer Chan, developed what's called stress in the dish, where we could take mouse cells, these are not human, we could collect the EVs, do all the pharmacology and the validation of following stress, and ask about how would you build a better sperm? What is it that the EVs are communicating? What functionally does it change about the sperm? Okay, 2 things you need to remember about sperm quickly. They are really 2 things. They are dad's DNA and then the motor to get dad's DNA to the egg. That's really all they are. So if you were going to build a better sperm, what would you focus on? The motor. You would focus on the motor, right? So what Nicole Moon, an MSTP graduate student in the lab, focused on, if you take those sperm from a mouse and incubate them with EVs from the culture dish of the stress, right? Different times, different exposures, et cetera. What has she learned? Well, what she learned is that it changes the mitochondrial respiration of the sperm, ultimately increasing its motility. And that is super exciting because if you have millions of sperm, it's a race as to who gets there. One sperm fertilizes, but a bunch of sperm are involved in that process. If they're all carrying a signal and they get there fastest, right? They're carrying a signal that says, we had stress in the environment, Probably we should increase fecundity. We probably should build back up our population that was just devastated by pandemic, devastated by famine, devastated by war, right? So it makes sense evolutionarily that there should be this signal. What does it mean to humans? Well, we are now repeating this on campus here. Hopefully you've seen flyers around with little sperm on them, or maybe you saw different things happening on campus, the sperm costumes out there. This is it. This is the study. We're doing all kinds of different assessments, which I don't have time to talk about. This is another grant that Neil and I have from NICHD that's asking these questions. We have preclinical studies asking about doing injections in mouse embryos. What does it mean when the small non-coding RNA content changes? What does that change for us about the brain? That's what we're really interested in. Okay, so I'm going to show you some data that Nicole just got. Super exciting, looking at human sperm, prior perceived stress, modeling very similar to what we did at Penn, and how perceived stress changes sperm function over time at 0, 1 month, 2 months, 3 months. The exact same thing we see in the mouse. And the exact same timing, right? We go out to 6 months. The exact same timing that we saw in the study we did at Penn. So super great to replicate the timing. Lots and lots of work to do. What does it mean for the embryo? What does it mean for development? All those questions, I get that for sure, but super exciting. So in summary, I hope that you see now the opportunities, especially around biomarkers. These happen to be extracellular vesicles, all kinds of questions. We're going to start a new study on campus with some of my psychiatry colleagues to ask in healthy, normal young adults, if you come in and we get you all relaxed and we get a blood sample and a saliva sample, do the EVs in saliva in any way replicate things we can see in blood? Because that opens up lots of opportunities you can imagine if you can just collect saliva versus having to collect blood. And then stress you and then to get another sample. Right, so that's the studies we're going to be ongoing. Lots and lots of questions to ask. Happy to answer any, whether it's here or you're thinking about it later in your clinic. Shoot me an email. We're hoping to really get started on ways to bank a lot of samples collected from different tissues and ask questions, collaborating with our colleagues in pediatrics who do sheep models. You can do such cool things that you can't do in a mouse or a human, where they can actually get samples right from the placenta, right? You can do the intrauterine artery and vein and the fetal artery and vein and do all kinds of glucose changes, cortisol. Really exciting to help us understand maternal and fetal health. Anyway, those are my outcomes. I need to thank the incredible lab members, which are bolded here on your left, who have done all this work over the last 5-10 years that have been involved in these studies. Many of them are assistant professors now out running their own labs. And of course, the Grady Trauma Project that we collaborated with Tanya's group, who are now at Wayne State in Detroit, building another understanding of trauma. As I said, follow us on Twitter. We do a lot of things working up different programs in the community, hopefully a lot of things around the Aurora community, since we are in Aurora on our campus. Our NIH funding across 3 different NIH institutes, and I'm happy to take questions.
Thank you so much. That was brilliant. That was so exciting. I loved it. I think we have time for 1 or 2 questions. I know we have a lot of endocrinologists here.
So I'm not an endocrinologist. But I guess my question is, when you talk about like the different groups that you study, it seems like, you know, trauma and socioeconomic status seem to go hand in hand. But from my understanding, adverse childhood experiences can affect anybody regardless of their socioeconomic status. So how do your current research efforts sort of capture the population of people who maybe come from stable economic backgrounds, but still have experienced a significant amount of adversity?
Yes, so fantastic question. And in community type studies, a very complicated one. So all of our studies that involve recruitment, we are of course examining overall levels of trauma. The second aim actually of the last study I talked about, the sperm studies, actually assesses adverse childhood experiences as a way of examining a baseline as well as current exposure to adversity and trauma. This is why through psychiatry, doing all of that assessment. I think that in the study at Penn, because one subject had, so if anybody's familiar with the ACEs, right, these are accumulation of the Kaiser Permanente study focused on 10 specific types of adversity. It has become very clear that it was based off of an overall very white population in California where they identified these 10. There are now other ACE surveys that take into consideration people coming from different backgrounds. But at Penn, it was an overall very white cohort that participated in that study and an overall very non-existent ACE. I think we had one subject that had one ACE maybe. We did not recruit for that. That's just the way that it played out. In hindsight, not even thinking about that as a factor. Thankfully, Neil, being very thought-provoking and forward-thinking, had captured all of that information. The second aim of our current grant is actually using that as a factor. So the first aim is using students on campus. You have to be a student to participate in aim 1. Aim 2 is you cannot be a student, so it's a slightly older population, taking into consideration of the ACEs scale and diversity on that socioeconomic background. So it's a great question. It's an important one to assess, I think, in all studies. How is that? How are you controlling for it as a factor? And how are you studying it as a factor? Yeah, and across the lifespan. Yeah, very important question.
Yeah, it was a great question. Thank you. We are right at 1 o'clock. So I'll say if there are other questions for Dr. Bale, please come to the front of the room. And Dr. Bale, thank you for being here.